Hello. Uh, want to go over a couple of quick things about the passive versus active transport activity you're going to be doing. So let's go ahead and quickly take a look at it. Your paper should look kind of like that one over there. The first thing is just a kind of a reading comprehension thing. Here's how I approach passages like this, and I think this is a good idea, especially if you're taking a test like this, right? Read the questions first before you read the big paragraph, because if you know what questions you're going to be asked, your brain will know what to look for in the reading. So that would be a quick suggestion. Maybe it'll help a little bit. Okay. The second part is simply a graphic. All you're going to be doing is taking these words and putting them in the correct box. Okay. One quick note here: the reading will help you with that, right? So will your notes, and you're welcome to use your notes. Okay. You're welcome to use the notes you just took along with your notes on passive transport. This is where things get a little bumpy, and I happen to know that there are questions like this on the test, so I wanted to give us as much practice as possible. What you have here, similar to our dialysis lab, are little cells inside a liquid. What you're going to do is use blue arrows to show which direction the concentration gradient is for water, so what direction water is going to move, red arrows for salt, and green arrows for glucose. And you'll notice some of them don't have glucose, some of them do, so just keep that in mind. Here's how you would think about this. Remember, for passive transport, things are going to go from an area of high concentration to an area of low concentration. So let's take the first one, for example. Here, the cell has 90% water. The beaker has 80% water. Water is going to go from high to low, so that means it's going to go from the cell into the beaker. So I would draw my blue arrow from the cell into the beaker, going out. That being said, salt has 10% in the cell, 20% in the beaker, which means salt's concentration is higher outside than in. It's going to go from high to low, so I would draw my red arrow from the beaker into the cell, because that's the direction salt would move. Okay, so keep in mind you want to go from high to low, so to identify what's high and what's low. All right, this next one asks you to look at specimens. I'm going to show you those right now. All right, the first one says salt ocean water. The next one says fresh water, and the next one says distilled water. So we're looking at a water plant, Elodea. We've all looked at that in the microscope, and this is kind of what we saw when we looked at it under our microscope. Okay, so this is Elodea under fresh water, isotonic. Notice how you have the cell wall, the cell membrane, you have all of this stuff sort of evenly dispersed within a cell. Okay? Ocean water is a hypertonic solution. So if we were to look at a hypertonic solution, in other words, we take this Elodea and we soak it in salt water, it would end up looking like that. Okay? If you look here, notice how the cell membrane has closed in on itself because all that water has gone out. Remember, hypertonic, we're going to lose water. So what's happened is all the contents of the cell has just kind of sucked in on each other because the water has evacuated. The water has left, okay? Because salt water, in this case ocean water, is hypertonic to freshwater cells, right? So the other option would be distilled water. Now, distilled water is 100% pure water, all right? So it's a concentration of 100% pure water. This is what it would look like, right? So this is hypotonic. Notice how the cell is kind of swollen up and kind of swollen with water, right? You'll notice how much it's bowing, like it's really fatter than even the cells in isotonic. What's happened there is water has gone in. Okay, so let's take a look at those for a second. If the teacher needs to pause, you're welcome to do that. Okay, if you guys need just a second. All right, the last part is for you to describe what's going on. If you recall in our lecture, this should be pretty easy. Okay, so good luck with this. Remember, your study guide is due for homework, so those are the questions after active transport, so part 4 of your study guide. And this assignment, they're due for homework. So make sure you have those ready upon return. I look forward to seeing you when I get back. Bye.